Hi everyone, thank you for tuning in for the lecture on Chapter 9, Microbial Disease Transmission. In this lecture, we're going to be talking about the 3 types of disease outbreaks: endemic, epidemic, and pandemic. We'll also be talking about various methods of disease transmission on how diseases are spread. Also factors relating to pathogenicity, and then lastly, we'll wrap up with non-specific host defenses in humans. It's going to be a wild ride. We have already established that there's millions of bacteria that are on us and in us at all times, and most of them are helpful. And, we find them in our soil and in other various parts of the world, and they really do a lot to help us out. And without them, we would cease to exist. However, there are a handful of them that are pathogenic to humans, and this is when it becomes a problem, right? And so those bacteria or microorganisms per se that can affect humans, they can come from nearly every member of the classification model, whether it is a virus or a bacteria, a viron, any of those things, prions, right, can definitely cause diseases in humans. And those are the ones that we're the most concerned with as we continue our journey through the amazing and maniacal world of microbiology and microbial disease transmission. The study of epidemiology looks at the distribution and determinants of health-related states or events. So if there are areas of the world where there are specific diseases, like that's the distribution, right? Is it happening in Europe? Is it happening in the United States? Is it happening all over the world? What is the distribution? And the determinants. What events specifically lead to these diseases occurring in populations around the world? So when epidemiologists do their thing, there's two different ways they study these diseases. They use descriptive studies and they use analytical studies. And when we're talking about identifying the determinants of a disease, we're looking at things like social and economic environment. What is the socioeconomic status of those people? What is the physical environment? Do they have clean drinking water? Do they have adequate shelter? And the individual characteristics of the population and what their behaviors are. Do they have higher risky types of behavior associated with that population? Is there gender differences between men and women? And what does the genetic situation look like as far as heredity and inherited traits, and how do they play a role? And then their personal habits and coping mechanisms. Do they drink? Do they smoke? Do they exercise? Do they use drugs? IV drugs? Those kinds of things, depending on what their behaviors are, could give us a clue of the determinants of so-called disease, whatever that disease is. We classify disease outbreaks in 3 ways. 1 as an endemic, 2 as a pandemic, or 3 as an epidemic. So an endemic is occurring constantly on an ongoing basis in a small localized area. The involvement is a small number of people, but it never really goes away, right? It never really gets eradicated. An example of this that the book gives is histoplasmosis, which is caused by Histoplasma capsulatum, and it is endemically associated with the United States. And an epidemic is going to be a disease that occurs with a greater than normal incidence of reported cases. All right, so we're kind of seeing a normal line of cases, and then all of a sudden we see them shoot up for some reason. All right, the percentage of the population that is susceptible is going to be eventually exhausted. So it's going to be a quick up and then a quick downturn. This could occur in a population that has never been exposed to a particular pathogen. This might happen when, as healthcare workers, we're traveling to a different country to help out, and the indigenous people there have been exposed to that flora that is there, but we may not have been. We didn't grow up with that, right? So we may be at a higher risk of contracting some sort of disease that the endemic people or the indigenous people may be immune to, right? Some other epidemic diseases of the past include bubonic plague, diphtheria, polio, scarlet fever, and smallpox. And now we're getting worried about West Nile virus, Lyme disease, malaria, pertussis, measles, polio, things that we thought we had eradicated, and individuals now are not getting vaccinated for it. And so now we're seeing these cases more frequently. And then lastly, foodborne infections like norovirus and cyclospora and listeriosis. And then lastly, we have pandemic diseases. Pandemic outbreaks, like the coronavirus, are worldwide events. And right now we are in one of those worldwide events. And we are a global society. We have the means to jump on a plane or on a boat and go to another country fairly easily, right? And because of that, that exacerbates the situation and leaves us more vulnerable to these pandemic events. A pathogen is anything that is capable of causing a disease. Now, while most microbes are non-pathogenic, and we've established that, even those that aren't pathogenic can become harmful if they gain access to tissues where they don't belong, or if they have some sort of genetic mutation, right? Like we've seen can happen. Some microbes, on the other hand, on the other side of the spectrum, are extremely dangerous under any circumstances to us or plants or to animals. And the ability of these pathogens to invade a host, that is called its virulence. Now, sometimes pathogenicity and virulence are used interchangeably, but virulence actually means the degree to which a pathogen is pathogenic, right? How strong is it, right? How virulent is it? Some microbes, of course, are more virulent than others, and are classified as infectious diseases, which means they result from an invasion and growth of a microbial colony. Communicable diseases are ones that are easily transferred from person to person. Conjunctivitis is a really communicable disease. And contagious disease is a communicable disease that can be transmitted very easily, like conjunctivitis was the example that I just used. So virulence factors, what are some of those? Well, colonization within the host. What tissues do they prefer? The respiratory tract, the GI tract, the nervous system, those kinds of things. The nutritional support, can they get the nutritional support that they need from the environment that they're in? They're going to look for that environment that is most conducive to their growth. Their ability to also invade and take down the host's immune system, that's going to contribute to its virulence. And then can it get in and can it get out of its host? Does it have a portal of entry? And that's kind of where we come to the discussion of pathogen reservoirs. And there are 5 main types of these. And the First one is called a human carrier. A human carrier is someone who carries the pathogen but is asymptomatic, so they might not even know that they have it. HPV is a good example of this. There's also been discussion that there are carriers for the COVID virus. Incubatory carriers are those that can transmit the pathogen during the incubation period. So this is that lag phase that we had talked about before, where, you know, there really isn't any bacteria growing, and then it starts to grow, but we haven't experienced symptoms yet. It's incubating. The convalescent carrier harbors a pathogen and can transmit it while recovering from the disease. So the convalescence period is when we're recovering. Our symptoms are tapering off, we're feeling a lot better, we're in that stationary phase or entering the death phase of the cycle. And cholera is an example of that one, where even after several months after the individual isn't sick, they can still transmit that pathogen. Active carriers are those that have recovered from the disease but harbor the pathogen forever. They've recovered, but they harbor it forever. And then passive carriers, they harbor pathogens without ever having had the disease. And human carriers often spread staph infections, hepatitis, dysentery, and STDs. A good example that the book gives, the most well-known one, was Typhoid Mary. Now there are also inanimate reservoirs and vectors. So vectors are like our mosquitoes, our ticks, our mites, our flies. They're a little living thing that, you know, hop from host to host and gladly transmit whatever pathogen they may be carrying. Inanimate reservoirs are things that aren't alive. And in the hospital, this could be any surface that you can get your eyes on. Can be considered an inanimate reservoir, and we also refer to that as a fomite. So it's anything that's not living. That does include the air, soil, food, and fluids, such as water and milk, and then any of those surfaces, contact surfaces in the healthcare setting. There are 4 main methods of transmission for these pathogenic microbes. And the first one is direct contact with an infected person. If you are administering care to someone that is infected, then if you touch them, you could be at risk of getting that disease. Sneezing is also an example of direct contact, as is secretion contact. You come into contact with their body fluids, and blood and body fluid contact. So this could be sharing of needles. For us in the operating room, Sharp's injuries, right? We are in direct contact with infected people. So we are going to be touching them. We are going to be exposed to their blood and their body fluids, and we are going to be using sharp things that have come into contact with their blood and body fluids. So we have to be really careful and make sure we use the best technique and safety practices. Indirect contact is when there's an infected individual that contaminates a fomite or food or something like that, right? Like hospital bedding, equipment, supplies, and then for food, hepatitis A is a common one. Contact with carriers. So these can be human, incubatory, convalescent, active, passive. We talked about all of those already. And then lastly, contact with animal or insect carriers. As far as Sharp's injuries that occur in the operating room, I found this little chart put together by the CDC, and it pertains to devices involved in percutaneous injuries. So Sharp's injuries specifically, And if you look at this big blue area on this pie chart, you will see that 59% of injuries come from hollow bore needles. This is the worst because hollow bore needles stay wet inside and bacteria likes wet. And so it is going to live inside there longer, and getting stuck with a dirty hypodermic needle is not a good situation. 59% of injuries come from that. Another 19% come from solid sharps injuries, like the suture needles. And then 8% is other, 7% from scalpel injuries, and then 2% from glass injuries. So we do work with like glass vials, and we have to snap the lids of those. And so glass injury could occur for us as well. And, you know, definitely scalpel injury is a possibility because we are passing those scalpels frequently throughout the procedure. Without a portal of entry, pathogens cannot be pathogenic. But there are several portals of entry that we're going to talk about. And the first one is the skin. So we have had prior discussions about the skin, and we said that the skin is the body's first line of defense. It's very difficult for microbes to penetrate intact, unbroken skin. However, there are a couple ways that they still might get in. They could creep in through sweat glands, pores. Some actually grow on the skin and under the nails, like specific type of fungi. And then there are some parasites, hookworms, that can penetrate the skin and become embedded there, which is very gross. The parenteral route means through the skin or beneath the skin. So this would allow for those pathogens to infect underlying tissues. And this is going to happen when there's a wound, an injection, a cut, or a bite. And remember, in surgery, all of our patients leave with wounds for the most part, unless we are performing a procedure that uses a natural opening, like the mouth or the nose or the rectum or the urethral meatus, those kinds of things. And so these breaks in the skin are going to allow those pathogens to directly invade the tissues that lie deep to the skin. Mucous membranes. The easiest way for pathogens to invade is through the nose or mouth. Okay, so the respiratory tract or the GI tract. And through the nose and mouth is typically through moisture droplets. However, when we're talking about the GI tract, there are some microbes that can enter through contaminated food and water. And while our very acidic environment of the stomach really protects us from a lot of those microbes, there are some that do love that acidic environment, and those are the ones that are going to be able to survive and are more virulent from that aspect. And so if they do survive, they're going to get eliminated in the waste or in the feces, right? And then it can then be transmitted to other people. Some examples of that is hepatitis A, dysentery, cholera, typhoid, and giardia. Some bacteria or microbes, on the other hand, are very selective, and they can only proliferate if they have a specific route of entry. And an example is the bacteria that is responsible for causing typhoid fever, which is Salmonella typhi. When it enters the body through the mouth via swallowing, then it can do its thing. But when it's placed on the surface of the skin, nothing happens. Yersinia pestis, on the other hand, is non-selective, and there are various portals of entry through which it can enter, proliferate, and do its thing. And then lastly, attachment to host tissues. A lot of these microbes, they produce some sticky, sugary substances on the surface of their cells, and this allows them to adhere to other surfaces. Strep mutans is a common one that contributes to tooth decay, and so its adherens that it secretes allows it to attach to the teeth. And another example is E. coli. E. coli, the disease-causing strains have adhesins as well, and that allows them to attach to the surface of the small intestine. As if bacteria didn't have enough tricks up its sleeve, there are also other factors that contribute to its ability to invade its host. Those are enzymes, bacterial capsules, and cell wall chemical substances. So some bacteria secrete various enzymes which allow them to do their dirty work. Some bacteria secrete enzymes that cause lysis of various substances that the body or the host would secrete to try to block their virulence, like dissolving clots and intracellular substances. Some microbiologists call these invasins, and these are literally the chemicals that they produce, the enzymes that they produce that will allow them to invade tissues. And I have to tell you, Clostridium is by far got the most tricks up its sleeve of any bacteria that I have ever seen. And you know, it not only secretes something called hyaluronidase, which breaks down hyaluronic acid, which is responsible for holding our connective tissues together. You might even see it if you look at face products by L'Oreal or Olivole, that there is this, there are products that have this hyaluronic acid in there that help to firm up the skin, right? So Clostridium, that causes gas gangrene, secretes an enzyme that will break down that hyaluronic acid. They also secrete something that is called collagenase, and collagenase breaks down collagen, and these aspects of Clostridium allow it to burrow itself deeper and deeper and deeper into the tissues. And there's other bacteria that have other enzymes that they secrete as well, like leukocidin, which helps break down macrophages, phagocytes, neutrophils, leukocytes. So that part of our immune system that's going to come to the rescue to fight off this bacteria is actually producing an enzyme to retaliate. And then there are various kinases which lyse and dissolve clots, like streptokinase and staphylokinase. And then there's also hemolysin, which causes the lysis of erythrocytes. Now, we've been able to harness some of this weaponry that bacteria have and be able to use it to our advantage. So if we have an individual that has a clot, we can use some of these weapons like the kinases to break down and dissolve those blood clots. So we have been able to harness some of this, these activities and these enzymes so that we can use them to our advantage. Now, that some bacteria also form capsules, and this also helps to protect them further. We've also talked about how they, some can sporulate and make spores. Those that make capsules that help make them more virulent is the bacteria that cause flu, anthrax, and pneumonia. And then those cell wall chemical substances. A good example of a bacteria that uses chemical substances to increase its virulence is the bacteria that causes tuberculosis. And there are these chemical waxes that it secretes that helps it to resist the attacks from the immune system through phagocytosis. And then strep pyogenes also synthesizes an acid-resistant and heat-resistant protein. Fortunately, our body can develop antigen or antibodies to that protein, to that antigen, and help to destroy it. There are 3 ways that pathogens damage the host cell, and that is through direct damage, production of toxins, and immune reactions. Now, direct damage means that it's going to attach to the host cell and produce some sort of enzyme that can penetrate the cell, and then it can go ahead and damage the cell and begin producing some sort of toxin. And there are 2 different types of toxins. There are exotoxins and endotoxins. And exotoxins are predominantly enzymes, aka proteins, and they're produced by gram-positive bacteria predominantly. And the way that they work is that they are soluble in body fluids. And so once these exotoxins start floating around, they can be picked up by the lymphatic system and the circulatory system and transported throughout the body, leading to systemic toxemia, which is the entire body being infected by this pathogen. So exotoxins, like I said, are enzymes that are produced predominantly by gram-positive bacteria, and they're going to be produced inside the cell, and they're going to get transported outside of the cell. And there are 3 categories of them. There are neurotoxins that attack the nervous system, enterotoxins that focus on the GI tract, and cytotoxins that focus on destroying host cells and affecting their normal functions. And so these are seriously dangerous. Clostridium difficile, or C. diff, secretes 2 different exotoxins, and the good thing is that the human body can produce antibodies called antitoxins, and that can help to neutralize the toxins that are secreted by some bacteria. Vibrio cholera is another one that secretes cholera toxin. Clostridium botulinum, that causes botulism, can also secrete a neurotoxin, and this neurotoxin gets in the neuromuscular junction and does not allow acetylcholine to work there, and that leads to muscle paralysis. If you've ever heard of Botox, Botox is made from the Clostridium botulinum bacteria. Endotoxins, on the other hand, are made by a special membrane that's near the outside wall of the bacteria, and those are predominantly gram-negative bacteria that produce endotoxins. And so the endotoxin is going to be produced and held within the bacteria until it ruptures, and when it ruptures, then the endotoxin is going to go out into the tissues, and they typically cause the same things to happen, like fever, chills, muscle aches, and in some severe cases can lead to shock and septicemia. Endotoxins also can cause something called disseminated intravascular coagulation, DIC, which is an overstimulation of the coagulation proteins from that coagulation cascade that we've talked about before, which results in systemic clotting. However, the reverse effect soon happens because all of those clotting factors are used up. So then the person can't clot at all, and they're bleeding like crazy. All right, so you know, another negative aspect for endotoxins is that the body cannot produce anything to fight against them, no antibodies. And then let's talk about immune reactions. So these are the body's natural defenses, right? We've talked about the immune system and how the white blood cells are there, our army to protect us. So the bacteria can mess with the skin, the mucous membranes, phagocytes, complement proteins, iron, and cause inflammation, fever, and interrupt that natural process that our body has to protect itself. Just when we thought it couldn't get much scarier, now we have to talk about pathogenicity of viruses. And viruses get into the host by attaching to receptor sites of the host cell, and they have the ability to trick the cell into giving the virus access to its interior. And when it does, that's when it takes over the machinery of the cell. And these guys are really like pirates, I'm telling you. They commandeer the machinery, or they destroy it and break it down, or they damage it or mutate it or alter it. It's crazy how these viruses can have these effects on the host cell. And so how do they do that? What do they do? A good example is the virus that causes AIDS, or HIV, actually hides its attachment sites so that the body's immune system doesn't recognize the virus as being damaging, and then it allows it to attack the immune system. And viruses like HIV are examples of cell-specific viruses that only attack specific cells. So let's talk about the cytopathic effects of viral infection. Cytopathic effects are the effects that viruses have on the host cell. And they're often used in the lab as a means of diagnosing a viral infection. And so viruses either have cytocidal effects, which means they kill the cell, or they have non-cytocidal effects, which means they cause damage, but not death. And so something else that viruses do is that they can produce some different types of bodies that help us to identify them, and those are granules and inclusion bodies. And these vary in regards to shape or size or how they stain. Some are acidophilic, they stain with acid. Some are basophilic, they stain with a basic stain. But again, they help aid in the identification of the virus. An example of that would be Negri bodies that are present in those infected cells that with the rabies virus. Herpes and smallpox are also examples of those that have inclusion bodies. Other viruses can release enzymes. They will reprogram the lysosomes to release enzymes that damage or destroy the host cell's intracellular structures, and that will result in cell death. They can also disrupt other activities of the cell, such as inhibiting mitosis of the cell. Can also damage the chromosomes, cause mutations. They can inflict antigenic changes on the surface of the infected host cell. This can trigger changes in the body's immune system and its ability to destroy the cells with antibodies. There are also cancer-causing viruses, and these viruses alter the host cell and they become abnormal, right? That's cancer, proliferation of abnormal cells. And then the growth becomes unregulated because it's no longer recognized by the body, which would explain the abnormal, uncontrolled growth of those infected by those viruses. Now, previously in anatomy, we've talked about our non-specific host defense. Now, that is our innate system, right? When we're talking about our innate system, those are things that we're just born with, right? And one of those is our skin, right? And so our skin doesn't protect against a certain type of microbe or pathogen. It protects against them all, right? All invaders. And so the skin is really our first protection our first line of defense. It forms a barrier that protects our underlying tissues. But like I said before, there are little pores and sweat glands where bacteria could potentially get in. There are special enzymes in our sweat, however, that does break down gram-positive bacteria, so it's helping us out as well. Mucous membranes that line the respiratory tract, the GI tract, and the genitourinary tract also help us out. Even though they are a warm, wet environment where bacteria like to grow, in areas like the respiratory tract, there are these little cilia that are on the cells, and that helps to move that debris and dust and microbes up and out to be expelled from the body. The genitourinary tract gets flushed by the urine, and the urine is relatively acidic. So again, that prevents the colonization of microorganisms in the tract. Our stomach secretes hydrochloric acid, which is really strong acid with a pH of about 1, and most bacteria can't survive in that. However, there are a couple that can. C. botulinum and Staph aureus and Helicobacter pylori can all live there and flourish there. And the H. pylori contributes to the cause of stomach and duodenal ulcers, like I think we've talked about before. So that's like our first lines of defense there. And then we move into our second line of defense, which is our immune system. And our non-specific immune system deals with phagocytosis, and that is our cells that are the white blood cells. And so there's 5 types of blood cells: white blood cells, basophils, eosinophils, lymphocytes, monophytes, and neutrophils. And what happens when we get an infection is that white cell count goes up. So if it goes up over 10,000, then we're probably thinking along the lines of the person has an infection. Something to point out that eosinophils, they're specifically geared to produce toxins that fight parasites, such as helminths, which is interesting. But how phagocytosis occurs, that engulfing of invaders, it happens in 4 steps. The first one is called chemotaxis, and this is really where there is a chemical signal that attracts the phagocyte. And then the second step is adherence. So then it's going to attach to the surface of the invader. It's going to extend its little pseudopods and wrap itself around the invader and eventually engulf it on all sides. And then it's going to release some enzymes that will allow it to be broken down and digested. And digestion is the last part of that process. There are approximately 30 complement proteins, and these are proteins that bind to different antigen-antibody immune complexes, and they help to start that complement cascade, which is increasing the inflammatory response, allowing the antibodies to assist with the lysis or the breakdown of the antigen. It's going to help aid in neutralizing the toxins of some microbes and also attracts the phagocytes there, so responsible for chemotaxis. Iron is something that a lot of bacteria need to synthesize their exotoxins, so our white blood cells produce something called interleukin-1, and that is going to signal the liver to store oxygen, or to store iron, excuse me, so there isn't as much of it freely floating in the blood. And this is going to ramp up that inflammatory process. And remember, as we've discussed before, that inflammatory response, there's 4 signs to inflammation, and that is pain, heat, redness, and swelling. So the vessels are going to dilate at the site, and that's going to allow all of those cells to come to our aid, to our rescue. Phagocytosis is going to occur, and fever may also be our body's response, because that is going to increase the overall body temperature, and hopefully that's going to help to fight off some of those invaders as well. So that concludes our review of microbial disease and transmission. If you have any questions or comments or utter feelings of love for microbiology, save them for class.